We'll continue to the second speaker, Dr. Jill Webster. Dr. Jill Webster is a chief scientist in New Image International and New Image General. Please, the committee, please share the CV of Dr. Jill Webster. Okay, Dr. Jill Webster completed her BSc in the field of botany and zoology from University of Reading, UK. And then she completed PhD at immunology at University of London, UK. And currently she is as chief scientist at the New Image International and New Image General Nutrition at Auckland, New Zealand. Good morning, Dr. Jill. How are you doing?
Good morning.
Yeah, it's great to see you.
Yes.
Oh, good afternoon in New Zealand. Okay, what time it is now?
It's nearly 4 o'clock in the afternoon.
Okay, yeah. Here is almost 11
yes.
in the morning. So yeah, some time difference.
Yes.
Well, to cut the long story short, we would like to hear more about immunology aspect from your perspective as a scientist and also part of the company. So we hope that later on during the discussion, we can elaborate from what Dr. Austin have presented about, let's say, basic immunology and immunology related to COVID and research that you have conducted and product development probably in your company.
Yeah.
So the time is yours, 30 minutes for the duration of the presentation. Thank you.
Thank you very much for a wonderful introduction. And it's an amazing opportunity to join the team for this very auspicious and timely seminar. So yes, I come from a background in human translational immunology with a background in development of novel immunotherapeutic approaches. And I think my talk follows on very nicely from the previous presentation. So I'd like to speak about the potential of bovine colostrum as an oral immunotherapy for all aspects of COVID infection. And I think we need to see these approaches as adjuncts to any current vaccination protocols. As well as New Image, I'm chief scientist of the Institute of Colostrum Research, and just in brief, it's a foundation taking an initiative really to bring together colostrum-based research right from the bench, so preclinical discovery all the way through to clinical research, with really the goal to extrapolate everything that's been found for the benefit of mankind. So first, I'd like to give a brief background to bovine colostrum. So what is the definition? What's it actually composed of? And in that regard, really looking at the micronutrients, so the immune factors and all their functions for the immune system. So colostrum is bovine origin and it's harvested from milking one. And it's important to highlight that calf welfare is paramount. And we know this because if a calf doesn't receive colostrum within the first 12 hours of life.
Sorry, Dr. Jill, the slide has not been seen. It has not been shared. Could you perhaps repeat it a bit?
No, I'll try again. It did say screen sharing. I'm sorry.
Yeah, it's stated like that, but apparently no slides appear. Yet,
Let me try again. So I'm in PowerPoint. It says I'm screen sharing. So I'll stop share. I'll share screen again. Did you see the slides at all before I carry on?
No, I see your face at the moment.
Okay, I'm sorry. Right. Well.
Now it's okay. Now it's
okay. So my colleague just checked by text to check I knew how to screen share. It did say screen sharing. Right.
Yeah. So it say what is so unique about bovine colostrum, right?
Yeah. So exactly. We're going through the background. Okay. Just quickly, what's unique, and here we go. restart. Yeah, so we want to emphasize this milking one, the gold colostrum as we know it, is crucial to the life of the newborn. That's how important it is, because unlike human mammals, bovine calves are born without an immune system. So due to our milk production system, there's always leftover first milking. So there's colostrum left after that calf's had its immune nutrition. And the purpose for the calf, and this is what we wish to exploit, is it's extremely rich in a very diverse, in fact, the most diverse range of preformed immune actives, otherwise known as immunological proteins. And just to emphasize again, actually bovine colostrum's richer in diversity and abundance compared to human colostrum, because in humans we have placental transfer, especially of the immunoglobulins, whereas that doesn't happen in the bovine species. So I'd just like to go through some of the key functional immune clusters that are in colostrum that assist recipient immunity all through the passive transfer mechanism. So we've already heard about, and I think we're all aware of the importance of antibodies towards the disease target. So we have two classes of IgG1, and then we have the mucosal IgA, and then we have the primary initial response IgM. Also, uniquely in colostrum are very broadly active, non-specific antimicrobial proteins, some of which you might have come across. So these are lactoferrin, alpha-lactalbumin, beta-lactoglobulin, and then the enzyme lactoperoxidase, and also lysozyme. And there's another less well understood and well known low molecular weight fraction in colostrum, and these are called proline-rich peptides. So they're already preformed in the colostrum. They have a proline signaling domain. They play multiple roles in all developmental and physiological responses, but they're uniquely also broadly antimicrobial and immunostimulatory. Also in colostrum, we have some classic immunoregulatory cytokines. So the immunologists amongst you will be familiar with all of these, I'm sure. So we have transforming growth factor beta, interleukin-1 beta, interleukin-6, interleukin-10, and tumor necrosis factor. And all these work together in concert to mobilize a coherent, innate defense response. And key to this is the anti-inflammatory cytokine interleukin-10 that helps the immune response contract, which allows secondary immune activities to initiate. And important also is another cluster, which we call the growth factor cluster. So again, multiple proteins with very diverse biological functions, so insulin growth factor 1, epidermal growth factor, erythropoietin, and platelet-derived growth factors. So they're just some examples. So you can see it is a passive immune system that covers all bases, if you like, of an innate immune system. So it's also important to understand and appreciate that the bovine colostrum-derived immune regulatory proteins do have a high homology with the human immune system and its cellular receptors, which they're going to bind to. So it's well established that colostrum immunoglobulins function in the same manner as the human equivalent. And here in COVID situation, we're referring to their role to bind pathogens, but also importantly interact with the mucous membrane, which is the site of infection. So that's our gut, mouth, lung, oral cavity, etc. And we know that, as I just mentioned, that these immunoglobulins bind to the antibody receptors on immune cells that drive innate and adaptive immunity. You've just heard about all these immune subsets that are important. So we have B cells, dendritic cells, which are antigen-presenting cells, monocytes, macrophages, neutrophils, and natural killer cells. So all these cells can interact with antibody complexes and elicit a subsequent desirable immune response. So how come we're thinking about colostrum and the use of its naturally polyclonal antibody reactivity? So it's all about the lifestyle of the cow. So it's grass-fed, and these antibodies in colostrum are formed in response to natural exposure of a broad range of environmental pathogens. And it's all to do with them feeding on the plant, the grass, which is grown in the soil. So we've got two microbiomes, the soil microbiome and then the plant microbiome. So when the cow is feeding, it's naturally now being exposed to thousands and thousands of naturally arising organisms, some of which are our pathogens. So there's this cycle going of direct contact. And so when it has its calf and is making the colostrum, the mammary gland filters the immune actives out of the blood serum and then secretes it in the form of colostrum, so fat and some other macronutrients, but also its main goal also is to translate or transfer this immune system into the calf that's born with no immune system. So in this way, the calf gains immunity to its environment. So we're very interested in this cross-reactive immunoreactivity in bovine colostrum. And there's previous work that has formally demonstrated that colostrum does contain antibodies recognizing a whole wide range of microorganisms, some of which I said are human pathogens. And in this study, they looked at about 18 soil organisms and plant organisms and asked the question, you know, is this correct? The hypothesis that this natural exposure of the cow leads to autonomous protective antibody responses that we can exploit. So here's a list in this study of all the pathogens that were investigated for antibody reactivity. And you'll notice some human pathogens, E. coli, Helicobacter, P. acnes, staph, listeria, and the list goes on. So it's just an example. Reactivity in the colostrum was found against all of those pathogens, and I just wanted to show 3 examples of the nature of the findings. So these are all immunoassays, so looking at the IgG in the colostrum binding to a specific antigen from a specific pathogen. And we're looking at 3 groups. Non-immunized, whole colostrum, skim milk control, because obviously skim milk doesn't have antibodies in it. And the goal was to see if the non-immunized immune activity could be boosted by immunization with the pathogen. And the take-home messages were repeated across all of the organisms. So if we look at Staph aureus and focus on the gray bars, this is just native colostrum. You can see as you decrease the antibody titer, a nice dose response. And in fact, in here, the immunized group didn't even do as well. So we can see we can detect clearly in fresh, whole, natural colostrum antibodies that react with Staph aureus. Similarly, with E. coli, again, a nice reactivity. And then the yeast candida, again, we're able to measure a titer equivalent or greater than the titer in a vaccinated target. So this sets the scene for there being lots of opportunity for colostrum antibodies to treat disease. So moving now into how this relates to the COVID challenge. So COVID isn't new. I know we're just talking about it in the last 3 years, but it's been in humans for more than 15 years. And they're all human coronavirus B members. So we've already had a SARS, that wasn't a pandemic. Then there was MERS, which was the Middle Eastern variant. And now we have the CoV-2 and its variants, such as Omicron. So of course there's going to be more. And in fact, I was reading today, there's a sub-variant of Omicron being detected. I made a note, they're calling it BA2. So there's another one coming. And they're saying it's even more infectious than Omicron. So it keeps on going, doesn't it? So if we think about the tools in the COVID toolbox, there was a lot of hope when we had the pandemic. Lots of papers, lots of clinical trials, lots of news articles. But in fact, these effective tools in our toolbox, in my opinion, are diminishing. So the vaccines widely used around the globe actually don't generate sterilizing immunity. They're not mucosal. Especially the mRNA vaccines, they're not generating such a broad epitope antibody response as you might get with a whole protein-based vaccine. And it's important to note these are being developed, but they take longer to develop than the mRNA vaccines. There was a lot of effort put into determining whether the salvage therapy based on convalescent plasma offered an opportunity for salvaging patients in ICU. But in fact, it turned out that it wasn't clinically effective, and there was a lot of side effects, and we're not seeing much development in that space. But what we are seeing is companies trying to isolate monoclonal antibodies from those convalescent plasma and somehow make a donor-based monoclonal antibody infusion. But again, it's monoclonal, not polyclonal. We've seen testing of a lot of repurposed pharmaceutical compounds, which, as we know, many have failed to translate to approved off-label use. The most success we've seen is the use of general anti-inflammatory based drugs. As I just mentioned, it stands to reason monoclonal antibodies such as a hybridoma-based antibody therapy, it won't be a cocktail, and therefore it does naturally have limited scope for targeting the whole virus, and obviously it won't necessarily be able to support targeting of any escape variants. So how can we relate colostrum to both the COVID infection and also recovery from COVID? There's really four categories we need to think about all at once. So obviously the first is to defend and protect our barrier function, and in the previous talk, those were also highlighted. So these are our mucosal surfaces, so our lungs, upper respiratory tract, and also our gut. And that will obviously reduce likelihood of infection taking off. And then obviously, in terms of a therapeutic, we've got treatment and recovery. And this includes limiting and constraining the impact of a successful infection once it takes hold, and allow good resolution, promote recovery, and hopefully interface with the adaptive immune system very well to generate one's own protective, long-lived response. So I'd just like to show my schematic of how colostrum fits into all of these challenges. So, of course, it's a respiratory disease, but it's now well measured that actually the CoV-2 is fairly promiscuous because it binds to the ACE2 receptor, which is expressed on many of our body tissue cells, including our blood vessels. So it also infects the gastrointestinal system. So we think of it as a respiratory disease, but it's also a GI disease. And I just want to highlight, and the consequence of this is cytokine storms, which are systemic. And then because there's immune skewing through the cytokine storm, there's increased susceptibility to secondary infections. And that's been reported in the clinic a lot, that they have pneumonia, for example, because the lungs have been compromised. So we look at early stage protection, we're looking for immune protection for the oral cavity, upper airways, and gut. So key to this are our immunoglobulins, as I've described, all classes, especially IgA, and then the non-specific, collaboratively active antiviral proteins and peptides, again, that I mentioned. So the proline-rich peptides and the lactoferrin, etc. So if we look at what's required for boosting and activity at late stage protection, as I mentioned, we're really trying to limit the damaging inflammation. We're trying to mitigate the likelihood of opportunistic second infections, and increasingly recognized, and this is the next COVID disease, hopefully somehow reduce the likelihood of developing long COVID. And some important colostrum factors to this side of the equation are those tissue repair factors, those growth factors. The anti-inflammatory mediators, so those are the cytokines in interleukin-10 and actually TGF-beta are very important. And again, we now understand how polyclonal immunoglobulins actually play a positive role in restoring the health and the intactness of the mucosal barrier. So under inflammation, it opens up, and part of recovery is it needs to close up again. And finally, thinking about long-term damage, colostrum, and I'm going to show you some data, also promotes stem cell factors. So just summarizing what I said in that mechanism slide, we want to discuss the exploitation of cross-reactive antibodies, which are polyclonal in the colostrum. And we want to look at their ability to cross-react with coronavirus strains. And we want to consider all of the effective immunoglobulin subclasses, so IgG, A, and M. I mentioned it just before, but I think it's really important to really understand why polyclonal antibodies are more relevant than pharmaceutical monoclonal antibodies. So the point about a polyclonal antibody is it's actually multiple B-cell clone-derived immunoglobulins. So you end up with a diverse, heterogeneous antibody population. And if you consider one antigen, you're going to find antibodies that are all different, that react to different regions of the antigen. So you've got built-in diversity. With a monoclonal pharmaceutical antibody, it literally is only a single B-cell clone, so you can only ever get a single species of antibody back out. Of course, you can make cocktails, but it's never possible to replicate a natural polyclonal response. And the main point is, if you look at the same antigen, you've got an antibody that only recognizes one part. So polyclonal antibodies have built-in redundancy for the antigenic target, and in this case, we're talking about COVID. So back to the bovine and the exposure to their environment. So there's previous evidence that firmly demonstrates that bovine colostrum contains anti-coronavirus antibodies. And this is another immunoreactivity and ELISA assay looking at bovine coronavirus B strain. And you can see for 2 colostrum products, as we dilute it down, we see a nice dose response emerge. So yes, cows have an endogenous bovine coronavirus. It's clinically silent. So we took this finding and translating it into human. So what we've shown is that these bovine coronavirus reactive antibodies can also interact with human coronavirus strains. So I just mentioned cows have a silent coronavirus B family infection. It's almost constitutive in New Zealand and North American cows, for example. So what we can see here is a virus neutralization assay result using antibodies purified from colostrum. And this is done in conjunction with our colleagues at the antiviral division at Utah State University, who run an antiviral testing and infectious testing containment facility. So we're just looking at a generic representation of the parent of our current COVID, which is coronavirus B strain. Here we can see, as we reduce the concentration of the antibody, we go from high neutralization and again see a dose response. So here we're looking at the antibody to bind to the virus, and therefore it can't infect the cells. So these are in vitro antiviral neutralization assays. We did the same experiment with a SARS-CoV-1 isolate. So again, high inhibition of infection with the highest antibody concentration, and as we decrease the antibody concentration, we see a nice dose response. At the beginning of the CoV-2 outbreak, the Utah University got their hands on a single human SARS-CoV-2 isolate, so one of the original ones. And again, we can show we have effective neutralization with our purified antibodies. And at the same time, in the same way of thought, I'll quickly share this, that if you look at the natural exposure of cow and their viruses, we did some studies with some other viruses of human health interest, so some tropical viruses like yellow fever and West Nile, another common one, respiratory syncytial virus. We had an outbreak in New Zealand last year, and then adenovirus and measles. So again, we're looking at the opposite scale of the previous graphs. Now we're looking at 100% infection and zero. And if we include our antibodies, you can see strongly neutralizing activity with RSV and again yellow fever. So the infectivity is only under 20%. And then we have relatively different levels of subsequent neutralizing activity for your West Nile and SARS. And then here we have also neutralizing activity for adenovirus and measles. And the important point is cows have their bovine counterparts of all of these viruses. So what about immune modulatory activity? We want to exploit these colostrum established immune signaling networks that we know deliver immune actives to the gut and our mucosal surfaces. And we want to exploit the presence of growth and repair factors that can reestablish mucosal and gut barrier function. So we've done some research on this low molecular weight peptide fraction of colostrum, and we've shown it has potent immune stimulatory activity for key cytokines involved in fighting infection. So this is an in vitro assay using human peripheral blood immune cells being cultured with different concentrations of the low molecular weight colostrum extract. And then we're doing immunoassays for key indicator cytokines using ELISA. So here we have our key innate antiviral cytokine called Interferon Gamma, and we're comparing two samples from different colostrum streams. So we're doing a duplicate, but that's of the raw material as well. And you can see that interferon gamma is produced in a dose-responsive manner with both our low molecular weight samples. If we look at interleukin-6, this is one of the master innate immunoregulatory cytokines. It conducts effective and efficient innate and adaptive immunity. Again, with our two samples, we can see a nice dose response and induction of interleukin-6. Moving on for the indicators of colostrum stimulated regenerative and repair. So there's a couple of key factors that report on that. One is granulocyte colony stimulating factor that drives bone marrow hematopoiesis. It replenishes and contributes to increased immune cell numbers, which are required during a vaccine response or actually during an infection. So here again, our two samples show actually a very high GCSF response. And then another important one is vascular endothelial growth factor. So that's obviously important in the vascular system, but also it's important in integrity of our barrier functions. And this is mislabeled, but this is a vascular endothelial growth factor response, again, across the two samples. So thinking a bit more about the recovery and repair wing during the COVID infection, I mentioned earlier how stem cells play an important role in replenishing the immune system, but also in driving the cell regenerative network. And we've undertaken some animal studies to look into this. So first, looking at hematopoietic stem cell numbers in peripheral blood. So this is an oral gavage mouse study, looking at colostrum, or one of our fortified colostrums. So here we're looking at the absolute number of hematopoietic stem cells in the blood, cells per microliter, performed by absolute counting and flow cytometry on unmanipulated whole blood. So compared to water gavage, all of our colostrum regimes at 14 days showed a huge statistically significant increase in the number of circulating hematopoietic stem cells. So we also looked at the mesenchymal stem cells, which are resident in our tissues and contribute to local tissue repair. Again, showing results now for mesenchymal absolute stem cell number in the microliter of blood. Again, compared to a water-fed control at 14 days, both of our colostrums again showed an enhancement of circulating mesenchymal stem cells. So how can colostrum help? It's been demonstrated, as I've shown you, to contain antibodies that recognize many natural pathogenic microbes that are in our environment. We think that colostrum offers broad protection against all airborne pathogens. In fact, shown you some of the evidence about its anti-inflammatory capacity, which we need to temper the cytokine storm, and also that we have known growth factors which can drive cellular repair. I'd like to now show some evidence from the globe, actually, because there's actually quite a few of like-minded thinkers out there trying to understand the therapeutic potential of bovine and its immunoglobulins for COVID intervention.